Hello, my name is Lindsey Biehler, and I'll be presenting on the quantification of constitutive innate immunity of songbirds against Salmonella. Salmonella is a significant zoonotic pathogen, and it is a gram-negative bacteria that can transmit between birds and humans. This figure shows the possible transmission of Salmonella from sick birds that eat at bird feeders to both humans and other birds at the bird feeders. This is done through the accidental ingestion of feces contaminated with bacteria, where either birds eating at the bird feeders can eat it, or humans can eat it via accidental ingestion from either touching the bird feeders or handling the sick bird. And Salmonella replicates intracellularly in the epithelial cells of the intestine. Clinical signs of the disease in birds include ruffled feathers, diarrhea, lethargy, with death shortly following symptoms. And wild birds act as both the reservoir and disseminator of the bacteria throughout the ecosystem. In 2020 to 2021, there was an outbreak of Salmonella that resulted in the mass die-off of songbirds. In 2021, the CDC reported 29 illnesses and 14 hospitalizations because of Salmonella linked to wild songbirds in the Pacific Northwest. This was in humans that handled either the sick birds or handled the contaminated bird feeders. This outbreak was related to the COVID-19 pandemic because with more people at home, they purchased more bird feeders and increased the number of bird feeders where Salmonella can be transmitted between the songbirds. And this is because people do not normally clean their bird feeders, and this allowed increased transmission between them. Finches readily occupy bird feeders where Salmonella can be transmitted, and from 1985 through 2004, 21.5 wild mortality events in perching birds involved Salmonella, with pine siskins being especially susceptible to Salmonella because pine siskins are frequently found dead near bird feeders during outbreaks. And the bottom picture here shows a pine siskin. More resilient finches may have superior constitutive innate immunity, which helps birds resist infection. And so we wanted to compare songbirds in terms of bactericidal activity to look for this. Our hypothesis was that bactericidal ability will differ between songbird species, and we predicted that spinous species commonly succumb to Salmonella infection. We predict that their ability to kill the bacteria in vitro will be worse than other finch species, with the pictures here showing a house finch on top and a lesser goldfinch on the bottom. Sampling was done on Fresno State campus with a combination of basket and ground traps. Our target species included house finches, pine siskins, and lesser goldfinches, with the house finches being sampled around Beetle Crossfield because they really like nesting in those cypress trees, and the pine siskins and goldfinches being sampled in campus. We did it around like the Rose Garden and the Keithis building. We also used a local Salmonella strain from a dead pine siskin for the assay. And we also did other measures that include banding and measuring the general morphological characteristics of the songbirds, as well as collecting more blood for later genetic characterization. This figure here shows the methods for the assay, with first blood being taken from the brachial vein of the birds, and then adding it to CO2 independent media that was used to maintain the leukocyte viability. 2 microliters of blood was needed for the assay, with 3 replicates happening for each bird. The blood was taken within 5 minutes of trapping the bird, and it was processed within 2 hours of initial blood collection. Then we added the diluted bacteria and TSP and allowed it to fully incubate for 18 hours. After this, we measured the absorbance at 600 nanometers using a nanodrop spectrometer. And we eventually calculated the antimicrobial activity with the question shown in the bottom with it being equal to 1 minus the absorbance of the sample divided by the absorbance of the positive control. And the positive control being the bacteria, PBS, and PSB. These incubation times were confirmed with Broker, and we used this to find the proportion of microbes killed in samples relative to the positive control. Our results are shown here in this figure, where it shows the percent Salmonella kill distributions for the lesser goldfinch, pine siskin, and the housefinch, with there being a clear difference between the housefinch and the other two, the lesser goldfinch and the pine siskin, who are both from the same genus, Spinus. The lesser goldfinch data is rather preliminary because we only caught 12 of them, but we did catch 24 of the pine siskins and 29 of the housefinch, and there is definitely a significant difference between those two. We also did a NOVA on the data set. We found the F value and the P values there. The data was also normally distributed and we tested. Previously, bacterial activity of housefinches had been measured in E. coli, though. And I couldn't find any information on pine siskins, lesser goldfinches, or other finches being used to look at the bactericidal activity using a BK assay. So our study is first to assess innate immunity against the pathogen that causes declines in their population. In conclusion, there's a significant difference between the bactericidal activity of Spinus species compared to house finches. And I was able to quantify their constitutive innate immunity function using the bacteria killing assay. This confirms that pine siskins are significantly worse at dealing with salmonella infections and are more susceptible to the bacteria. For my future research, my next logical step would be seeing if there's a relationship between bactericidal activity and genetic diversity of toll-like receptor 4, which is the receptor that recognizes and deals with gram-negative bacteria. And here are my references. Thank you.